Hello, everyone. It is my pleasure to have the opportunity to speak at this meeting. I'm Jia Tong from Beijing National Day School, and today I would like to present the poster of my research. I would like to first introduce the background and purpose of this study. So during the COVID-19 pandemic, in order to prevent the spreading of the coronavirus, we require frequent disinfection for communal facilities. And in the most common disinfectants we use, for example, hand sanitizers, surfactant is a very important active component. A surfactant is a surface active molecule which can reduce the surface tension of a liquid. A surfactant molecule has an amphoteric structure, which means it has a hydrophilic or water-like head and a hydrophobic or oil-like tail. So during the disinfection process, the surfactant molecule interacts with bacteria and viruses through a specific mechanism. So as we can see in this diagram, this graph shows the amphoteric structure of the surfactants and how it interacts with the surface of microbes. So since the bacteria cell membrane and the virus capsid carries charge, during the disinfection, the hydrophilic end of the surfactant molecule is first attracted by the surface of microbes, and then the hydrophobic tail of the surfactant molecule embeds into the bacterial membrane or virus capsid, so the microbes are killed. So in order to complete the experiments in a home lab, we used feasible and simplified models for substitute for the real microbes. We used the silica dioxide powder to simulate the bacteria because it carries a negative charge when dispersed in water, as we can see in the diagram below. And we used the calcite carbonate solid powder to simulate the virus surface because it carries positive charge when dispersed in water. Now I would like to introduce the method I used to complete the experiment. So in the preliminary stage of the experiment, I bought surfactants and the solid powder mentioned above. The surfactants I use are benzalkonium chloride, which is a cationic surfactant, sodium udecyl sulfate, which is an anionic surfactant, and primary alcohol ethoxylates, which is a non-ionic surfactant. And all three surfactants are soluble in water. In the following steps, I created the correlation graphs between the surfactant concentration and their refractive index, which is measured by a portable refractometer. For each surfactant, we created 6 samples with different concentration, thus different refractive index measured. Then for the data collection part, I took 10 grams of each surfactant sample and then add 10 grams of the silica or calcite solid powder and mix them thoroughly. So after 48 hours, we set this time to ensure that all the powder precipitate and thus will not affect the measurement of the final refractive index. So after 48 hours, I measured the refractive index of each remaining solution, and I can find the final concentration of each remaining solution by the correlation. So lastly, I calculated the adsorption of each sample and I calculated the samples, calculated the adsorption with a specific formula. Now I'd like to share my results of this study. So we applied 2 different adsorption models. The first one is the multi-layer adsorption model. As we can see in this diagram, the adsorption increases with the surfactant concentration because they are multi-layer of surfactant molecules attached to the solid powder. And the second is the monolayer adsorption model. We can see in this diagram that because there's only one layer of surfactant molecule attaching to the solid powder, the absorption will stop increasing once saturated. And the diagrams. The graph presented here shows the relationship between the absorption and the equilibrium concentration, which is also the final concentration of the remaining solution in each sample. So for the cationic surfactant with the silica powder, we can see that the absorption increases as the equilibrium concentration increases, and that fits to the our multi-layer absorption model. And the anionic surfactant absorption with the calcite powder shows that the absorption had no significant changes as the equilibrium concentration increases. So compared to the first graph, in the second graph, the trend line is almost horizontal, and the absorption does not change when the equilibrium concentration increases. So that fits to our monolayer absorption model. For the non-ionic surfactant absorption with the silica powder, we can see that the absorption also increases with the equilibrium concentration, so it also fits to our multi-layer absorption model. So from the results above, we're able to conclude a few points. So first, we established a valid home lab to simulate the surfactant microbe interaction during disinfection, and we're able to quantify the absorption by creating concentration refractive index correlations. We conclude that the cationic surfactant bacteria and non-ionic surfactant bacteria pairs undergo the multi-layer absorption, and the ionic surfactant virus pair undergoes monolayer absorption. So from comparing the different different graphs in our results, we conclude that the electrostatic attraction is a dominant factor of surfactant microbe absorption, because in this first graph here, the cationic surfactant interacts with the silica powder, which has a negative charge when dispersed in water. And what we doesn't show here is the group of the cationic surfactants with calcite powder, which carries both of them carries positive charge. When we tested this pair, we found that the refractive index is too low for the refractometer to measure. So that's why we didn't post the correlating graph here. So to achieve high efficiency disinfection, we highly recommend to use cationic surfactant containing disinfectants for bacteria and anionic surfactant containing disinfectant. We recommend an anionic surfactant containing disinfectant for virus. So for example, during the COVID-19 pandemic, we recommend to use the anionic surfactant containing disinfectants to eliminate coronavirus, to stop the spreading of it. So future work can focus on the dominant factor of mixed surfactant absorption, which we think there may be some surfactants that are feasible for killing both viruses and bacteria with higher efficiency. That's the end of my poster presentation. Thank you very much.
Thank you, Jiapeng. So let's see if our audiences have any questions. I'm checking. So basically, I do have a question. How many number of trials have you done for this experiment? I can see from your results graph, like each surfactant will have 3 dots there. Does it mean like you have done just three trials for the different surfactant experiment?
Well, actually, at the start of the experiment, I plan to test all 6 concentrations because I prepared 6 samples in the preliminary stage. However, when I measured their refractive index, I found out that like there is a certain range in which the refractometer is able to generate like accurate data. If the concentration went too low or too high, then the refractometer is not able to to present like accurate data or like accurate trend. So like I eliminated the range from the 6 concentrations to the like 3 concentrations.
Okay, I see. Thank you. And also, one thing caught my attention is that at the beginning, you mentioned like all this research has been done in a home lab setting. Can you tell us more about like how you set up the home lab? Because I do know like in China, home lab is not very popular, right? And how you get started with this idea is set up a home lab and how this experience is like.
Because I completed this research during the like July and August, and so it's in summer vacation, and because of the pandemic, I was not able to go to school and complete my research in the lab in school. So that's how. So I thought of establishing a home lab.
Yeah, that is very cool. Because I do know, like at Rapidamic, we have a lot of students setting up home labs, right? And do you want to like tell us a little bit of your experience in setting up home labs versus like doing experiments in, for example, in the real labs at school?
So I think the biggest difference between a home lab and a school lab is in home lab, I have to like prepare everything by myself, like including purchasing the surfactant or the like all the instruments.
Mm-hmm.
And in school lab, I can just use what's provided by the school.
Right.
Yeah.
But that's another experience as well, right? Setting up your own lab, that is super cool.
Yes.
Okay.
Yeah, actually, the idea of the home lab starts even before the decision of my topic of research. Yeah, because as I mentioned, I started this home lab because I was not able to go to school in summer because of the pandemic policy. So I decided I will present a topic that is related to the pandemic and to eliminate the spreading of virus.
Mm-hmm. Sounds great. Thank you, Taco.